Hello, my name is Cuauhtémoc Solis. My team is going to talk about photosynthesis and cellular respiration, also fermentation, and it's composed by Leslie Tapia Reyes, Camila Rodríguez Bañuelos, Valeria Maciel Ramírez, Elizabeth Stephanie Hernández, Ana María Rosa Argüelles, and me. So we're going to start with the next slide.
So first we're going to talk about photosynthesis, that it is a process by which plants use sunlight, water, and carbon dioxide to create oxygen and energy in the form of sugar.
The steps of the photosynthesis are: step 1, it is during the process of photosynthesis, carbon dioxide enters through the stomata, water is absorbed by the root hairs from the solid and is carried to the leaves. Through the system vessels, chlorophyll 2 absorbs the light energy from the sun to split water molecules into hydrogen and oxygen. The step 2 is the hydrogen from water molecules and carbon dioxide absorbed from the air and used in the production of glucose. Furthermore, oxygen is liberated out into the atmosphere through the leaves as a waste product. These are the most important steps. And then we see step 3, that the glucose is a source of food for plants that provide energy for growth and development, while the rest is stored in the roots, leaves, and fruits for their later use. And then last, pigments are other fundamental cellular components of the photosynthesis. They are the molecules that impart color, and they absorb light at some specific wavelength and reflect back to the unabsorbed light. All green plants mainly contain chlorophyll and chlorophyll B and carotenoids, which are present in the thylakoids of chloroplasts. It is primarily used to capture light energy.
Now, photosynthesis takes place in two stages: the light-dependent reactions and the light-independent reactions. The light-dependent reactions require sunlight. Energy from sunlight is absorbed by chlorophyll and converted into stored chemical energy. Reactions take place in the thylakoid membranes in the granule within the chloroplast. And in the light-independent reactions, the energized electrons from the light-dependent reactions provide the energy to form carbohydrates from carbon dioxide molecules. Although these reactions do not use light as a reactant, they require the products of the light-dependent reactions to function. Now, the enzymes of the photosynthesis, one example is a Rubisco, that it is an enzyme involved in the first major step of carbon fixation, a process by which the atmospheric carbon dioxide is converted by plants and other photosynthetic organisms to energy molecules, such as glucose. And the proteins of the photosynthesis, there are four, which are the photosystem 1, the cytochrome B6F, the photosystem 2, and the ATP synthase.
Cellular respiration is another type of making energy in the living things. The first step to make cellular respiration is the glycolysis. In glycolysis, glucose, 6-carbon sugar molecule, undergoes of a series of chemical transformations. In the end, it turns into two molecules, pyruvate and a 3-carbon organic molecule. In these reactions, ATP and AD is converted to NADH. Then comes the pyruvate oxidation. Each pyruvate from glycolysis travels to the mitochondrial matrix in its innermost compartment of mitochondria. There, pyruvate turns into a 2-carbon molecule attached to a coenzyme A known as acetyl-CoA. In this process, the carbon dioxide is released and NADH is obtained. Then, the third step is the citric acid cycle. The acetyl-CoA obtained in the previous step combines with a 4-carbon molecule and goes through a cycle of reactions to finally regenerate to initial 4-carbon molecule. In this process, ATP, NADH, and FADH2 are generated and CO2 is released. And finally, the oxidative phosphorylation. The NADH and FADH2 produced in previous steps deposit their electrons in the electron transport chain and return to their empty form, which is NADH plus and FAD. The movement of the electrons down the chain releases energy that is used to pump protons out of the matrix and form gradient. The protons flow back into the matrix through an enzyme called ATP synthase to make ATP. And then the electron transport change, oxygen, receives the electron and picks up protons from the medium from water. The image right there is explaining all the steps I already said.
And the fermentation is a chemical process where is it breaking down of molecules into simpler compounds to produce substance that can be used in making chemical energy. This process does not use oxygen like the anaerobic respiration, but is different because the fermentation skips the electron transport chain system. The following enzymes are used for fermentation. One is the enzyme amylase, catalyzes the digestion of carbohydrates. It is hydrolysis reaction to digest starch or glycogen and form simple sugars, and the other enzyme is the glucoside that catalyzes the hydrolysis of glucoses that form a bond of the branching points of the glycogen. In the fermentation, the energy net gain is 2 ATP molecules glucose molecule.
So there are two types of fermentation. The first one is the alcoholic fermentation, which is done by yeast that helps in the process of making bread and making alcoholic beverages. The first step is when glycolysis happens. That means that glycose is converted into 2-pyruvate, 2-NADH, and 2-ATP. Then the 2-pyruvate produces carbon dioxide and 2-ethanol, which is alcohol. And then the 2-acetaldehyde oxidizes NADH to NAD+, and in this way the process can start all over again. Then the other type of fermentation is the lactic acid fermentation, which is done by the muscle cells, for example, in the process of making cheese and yogurt. The first step, as the other one, is when glycolysis happens. This means that, glucose is converted into 2-pyruvate, 2-NADH, and 2-ATP. Then the pyruvate produces lactate, and then the pyruvate oxidizes NADH to NAD+. And then this process can start all over again.
Here we have a comparative table between the photosynthesis, cellular respiration, and fermentation. In the photosynthesis, it occurs in chloroplasts. The process is used by plants and other organisms. It converts light energy into chemical energy, and it's divided into light-dependent and light-independent reactions. Its energy is stored, and it has 18 ATP. In the cellular respiration, it occurs in the mitochondria. Its process and reactions are used by cells. It converts chemical energy from oxygen molecules to ATP and then releases waste products. The energy is released and it has 38 ATP. And in fermentation, it occurs in cytoplasms. Process is used by microorganisms. It converts chemical changes in organic substrates through the actions of enzymes. The energy is released and it's divided in lactic acid and alcohol fermentation, and it has 2 ATPs. And we have examples in the bottom.
So thanks for your attention, and this is all of our presentation.